Thank you, Dr. Bau, for the kind introduction. So today I'll be telling you guys about carbon nanopipettes, specifically for advanced cellular probing and microinjection. So the motivation behind this research is single-cell studies. So anyone who's ever done biological research knows how polydispersed cells are. So within a given cell culture, cells have different sizes, different gene expression. They're very sensitive to their local environment. And so there's a growing need and interest in tools for studying cells at the single and subcellular level. And the problem that arises there is that single cells are on the order of tens of microns, and the subcellular level is on the order of the nanoscale, which introduces a lot of difficulty in manufacturing, manipulating, and taking measurements at this scale. It's also highly multidisciplinary, so there's kind of a crossover between a lot of different fields. My background's mechanical engineering, but you'll see hints of bioengineering, biology, chemistry, material science, nanotech, and neuroscience in the work that I've been doing. So the tool that is kind of the basis of all this work is the carbon nanopipette. So it was developed in our lab a few years ago. So the carbon nanopipette starts out as a pulled glass capillary. So we take a glass capillary, put it, shine a laser on it to heat it up to a high temperature, and then we apply a tension to pull it down to a very small size. And there's commercial equipment available to pull these pipettes, and the benefit is that you get a fluidic channel from the macro scale, so the pipettes are on the order of millimeter scale, and the pulled tip can get down to the nanoscale. So you have this continuous fluidic pathway from macro to nanoscale. We then take these pulled quartz capillaries, we put them into a chemical vapor deposition furnace at very high temperature. We flow through argon and methane, and at high temperature, the methane decomposes and deposits carbon all along the interior surface of the pipette. So there's actually a confinement effect that causes the carbon to only deposit on the inside surface of the pipette. And then once we have the carbon deposited on these pipettes, here you can see it's kind of darker after deposition, we can then put the tip into a wet hydrofluoric acid etch, which selectively etches the quartz. And the quartz thickness near the tip increases roughly linearly with the length along the pipette axis. And so you can control the amount of exposed carbon based on the amount of time that you etch these pipettes. And so based on the deposition times, you can have either solid pipettes where you completely fill the bore with carbon near the tip, or you can leave the fluidic channel open. And so this is very nice since we now have a continuous fluidic pathway from the macro scale to the nanoscale, so you can still do microinjection and things of that nature. We have also a continuous carbon layer from the distal end to the nanoscopic tip. And carbon is biocompatible, it's very strong, and it's also electrically conductive, meaning we can use carbon as a working electrode for electrochemical studies. So here's a couple SEM images showing both a hollow CNP, where the tip is still left open, and then we can also create these solid CNP tips with tip sizes ranging down to the nanoscale. And because it's template-based, we can control the dimensions and the geometry based on the pipette that we pull. So there's different parameters you can adjust. You can get down to the nanoscale, and we can control the amount of exposed carbon. So we have a lot of control, and it's batch fabricated, so you can make hundreds of these at once. You can also look to functionalize the surface with things like metals. For example, here we looked at depositing silver onto the CNP tip. So if you wanted to change the surface chemistry of your electrode, or if you wanted to functionalize it with, say, some antibody that there's a known surface chemistry that works with silver, but not with carbon, you can do things like that. So these are two examples of double pulse electroplating, and this is pulse current electroplating, just showing that we can use these different techniques at this scale to get a nice surface coverage of silver. So just a brief overview of the work that I've done. I've kind of lumped it into two categories. There's microinjection, so using the CNP for advanced microinjection techniques. And then we also kind of fall into a microelectrode category, and there is some overlap. So I'll be talking about a scheme we developed to detect cell membrane penetration with the CNP, a system that we're developing to automate the microinjection process, and a target biological study with fluorescent tRNA. And I'll also discuss fast-scan cyclic voltammetry, a collaboration that we started with UVA to detect neurotransmitters. So some background on microinjection. So it is exactly what it sounds like. We're injecting something with fluid at the microscale. So traditionally, microinjection has been done with larger cells. So this is a Drosophila embryo, a zebrafish embryo, frog oocyte, in vitro fertilization, which may be the most commonly seen example of microinjection. But if you're interested in injecting cells in recent years, people have started moving towards actually smaller cells. So these are all on the order of a couple hundred microns in dimension. When you move to mammalian cells and things like adherent cell cultures, you start looking at cells that are tens of microns in diameter, and the thickness is on the order of 1 to 5 microns at their thickest. So you're looking at injecting a very difficult target, a very small target. So the benefits of injecting single cells: number one, you get to work with very small volumes of fluid. So for example, you can work with loading the pipette only takes about 1 microliter of fluid, and the injection volume is on the order of picoliters to femtoliters, which is 10 to the minus 12. So if you have a very expensive reagent that you're trying to inject into cells, this is very nice since you can conserve the reagent instead of having to use several mils of it for a cell culture. We get single cell resolution. We get strong control over the composition that we're introducing into cells, since you're directly filling the pipette with what you want to introduce into cells. Other methods, such as electroporation, if materials have different electrophoretic mobilities, they'll enter the cell at different rates. And so it becomes a little bit more difficult and requires a little more finesse to get the desired composition into the cell. We can inject into the cytoplasm or the nucleus. So some transfection techniques have difficulty getting material to the nucleus, so that's nice. And it's adaptable to most cell types. So it's a physical technique. You're directly introducing material into a cell, so it's very adaptable to numerous types of mammalian cells. The drawbacks are that it's tedious and delicate. Like I said, it's a very small cell, it's a very small target, and even semi-automated methods that have come along have very poor success rates. And one of the reasons for the poor success rate is that there's a distinct lack of feedback to indicate if the pipette has penetrated a cell. So we're looking to lower the pipette into a region that's 1 to 5 microns thick, and you're not looking at it in the Z-plane. So it becomes, you have to basically see where the pipette actually contacts the cell membrane, and there's kind of a poor success rate. So some other groups have actually looked into this process of trying to detect cell membrane penetration. So Mike Charloux actually did it with CNPs several years back by measuring the membrane potential as the pipette crosses the cell membrane. And you can actually see the drop corresponding to the membrane potential of the cell. But the problem with this was there was no microinjection, so he did it with an empty CNP that was pressurized to stabilize the internal fluidic interface of the pipette. And the carbon is not very good for potentiometric measurements. It's very noisy. You need a very high impedance amplifier. Another group used SCCM type techniques, so scanning electrochemical microscopy, where they introduced a redox mediator into solution. So it's a salt that undergoes a redox reaction and you get current. And they get this curve as they're approaching a cell of the current drops due to the cell insulating and hindering the diffusion of this redox mediator to this micro nanoelectrode. And then when you enter the cell, the current plateaus. But this also used a solid electrode, no microinjection, and it required a redox mediator in solution. Another group actually used microinjection and used an electrical method to detect penetration, but they inserted an electrode directly into the injection fluid. And this creates limitations on the injection fluid, both the volume, because you need enough for it to be in contact with the electrode, and the type of solution. You typically need a very high concentration solution for the silver chloride electrode that they put into the pipette. So we're looking to use CNPs for microinjection. And the benefits that we have are improved durability and biocompatibility. We can achieve smaller dimensions, which means that they can be less invasive. It's compatible with existing equipment and technology, such as pumps, fittings, and amplifiers. So that's quite nice for people looking to adapt this technology. But most importantly, we have this carbon working electrode that's along the entire internal surface of the pipette. So we would like to use the carbon lining to electrically detect cell penetration during microinjection. So other electrodes for single-cell studies, a lot of people use patch electrodes, which is similar to the microinjection technique I described before, where you have a silver chloride electrode placed in a glass micropipette filled with salt solution. There's a very fast redox reaction of the silver and the chloride, which gives you a very stable potential and it's very good for recording potentials and applying currents. And then you have the constriction at the tip of the pipette, which localizes the measurement. And people do all sorts of patch clamp studies, generally contacting or inserting the electrode directly into cells. The other type of electrodes people use for single-cell studies are working electrodes. So it's typically an inert electrode material that's ideally polarizable that does not have a redox reaction of its own. It doesn't participate in the reactions, but it allows for electron transfer to take place to the species in solution. So instead of the electrode surface undergoing some reaction, you have some species in solution that gets oxidized or reduced. So these are generally ultra-microelectrodes in order to localize measurements to a single cell. But as I mentioned, these methods are not ideal for microinjection, so they're both dependent on Faradaic reactions, which are redox reactions. So if you look at, it's called a Randall circuit of the electrode interface, you have a double layer capacitance, a charge transfer, and Warburg impedance due to charge transfer to a species in solution, and this is a diffusive term. And then you have the solution resistance due to the conductivity of the solution. But without Faradaic reactions, without some redox mediator in solution, this impedance is extremely large. It's very difficult to pass currents through this impedance. And so you end up with basically a series RC circuit. And if you recall, for a capacitor, the current across capacitor is the capacitance times the time derivative of potential. So at steady state conditions, you pass no current. Essentially, it's a very high impedance interface. So when you're looking at doing cell studies, there's very few redox species in typical cell media. And adding chemicals to cell media complicates the studies that you may want to do. And we also want to maintain the fluidic channel for microinjection rather than needing to fill it with electrolyte for measurements, which makes the other patch clamp type electrode difficult to use. So we need a different mechanism to detect changes in the electrode environment. So because we have a capacitance, we wanted to look at AC methods. So we have a digital lock-in amplifier that's built into a patch clamp amplifier that we have in our lab. And we apply a 1 kilohertz perturbation potential, and then we record the current response. Then the lock-in amplifier uses Fourier methods, so it uses the orthogonality of the sine waves to isolate the current response only at the forcing frequency. So if you're applying a signal at a perturbation at 1 kilohertz, you only get the current signal at 1 kilohertz. And it creates essentially a low noise bandpass filter that's very, very accurate. And from this signal, you can then extract the complex impedance, and changes in the response can be attributed to changes in the equivalent capacitance or equivalent circuit of the system. So the basic premise of our scheme is that as the carbon tip crosses the cell membrane, changes in the local environment cause a distinct change in the impedance signal that we get. So again, we have this RC circuit. The impedance is basically the real part is due to the resistance, the series resistance. The imaginary part is due to the capacitance. And we can look at changes in these parameters as the CNP penetrates the cell. So you have differences both in extracellular versus intracellular composition, and then you also have that the electrical path now has to traverse the cellular membrane. So the lipid bilayer acts as a dielectric insulator. It has some conductance due to ion channels, but it's mostly acts as a capacitor. And we get this impedance increase and corresponding capacitance decrease, since they're related, upon cell penetration. We can also look at the effect of capillary rise in the pipettes to characterize what the effect of fluid in the pipette is going to be. So in this case, the capacitance of the internal surface will be proportional to the internal wetted area of the pipette. And this internal wetted area is a function of the balance between any pressure you're applying to the pipette and the Laplace pressure. And because the tip is conical, this yields a 1 over pressure squared relationship. And this plot here is just the equivalent capacitance versus the 1 over p squared, showing that we do indeed recover a linear relationship and that we can accurately capture the behavior of the pipette at different pressures with different injection solutions in there. So with that, we did some studies under 2 different modes of injection. So we have this plot here. The red curve and this lower axis is the equivalent capacitance, which is essentially the inverse of the imaginary impedance. We work with capacitance because there's electrochemically, there's known relationships between things like the solution composition and the capacitance. So it's very easy to work with and easy to analyze. And then the top blue curve is the real part of the impedance. And each of these arrows corresponds to the injection of a single cell under a continuous flow condition. So as you can see, we get a several picofarad, which is quite large drop in capacitance that allows us to very robustly detect penetration during microinjection. We can also look at the normalized capacitance change as a function of the solution concentration. Here we have it to the 1/2 power due to the trends we would expect from the literature. And we roughly get a increasing change in capacitance with increasing concentration. So we can characterize the magnitude of the signal and how it relates to the solution we're injecting into cells. We also looked at under pulsed injection conditions. So here we actually apply a pulse of pressure to inject fluid into the cell. So here we can detect, again, the red curve is capacitance, the blue curve is the real part of the impedance, essentially the series resistance. And we can look at the penetration events, and then we can also detect the pulse that we apply for injection. And there is some back pressure applied to the pipette to prevent clogging, so that leads to this kind of gradual approach to a steady state condition as the cell is slowly injected. And we also looked at this curve over time to show the stability. Another very nice thing about this technique is that the nuclear membrane acts as another membrane in series, and so we can actually determine whether we're injecting into the cytoplasm or nucleus based on the magnitude of the signal change. So we found that on average the injection into the nucleus was about 1.5 times greater signal change compared to the cytoplasm. So that gives us another tool we can work with for feedback. And so with this detection scheme, we started working towards an automated injection system. So a lot of this is work that a master's student that's been working in our lab has done, so Chi. He's been working with me for the past year or so. So we're developing a Matlab-based GUI that interfaces with the various microinjection components that we have. So there's a micromanipulator, an injection pump, and a CCD camera to look at the cells during injection. So we select points on the live camera feed to inject. We use a traveling salesman algorithm to calculate the shortest path. And then we have a piezoelectric manipulator that moves the pipette into position in order to inject the cell, and then the pump is triggered to inject. So the benefits, yeah.
Controlling flow rate or pressure?
The pump is pressure controlled. Yeah. So the flow rate is, at this scale, is linear with the pressure. So you can then either adjust the pressure to... The tip is nanoscale, so the flow rate through the tip is still going to be in the laminar regime. So we typically use pressure. You can adjust the injection time, so you can inject for, like, say, if you want to double the amount you're injecting, you can either double the pressure or double the injection time to compensate. But the benefits of this system are that we hope to incorporate this penetration detection to provide robust feedback, both of penetration, and then also you can look at changes in the pipette due to breakage or clogging, which show up as signal changes as well. And it also uses commercially available software and equipment, since it's Matlab-based and we're using standard Eppendorf equipment. So it would be very applicable to many labs. So this is what the system looks like. So we have this panel here for the live camera feed. So you can take videos and get your camera feed set up, various injection parameters. And then when you have your injection parameters, such as the speed at which you're penetrating the cells and the angle that you want to penetrate the cells at, you can then go into the injection mode. And here you can see the pipette traveling around to the different injection locations, lowering into the cells, and then injecting. So it's actually taken through the eyepiece of our microscope. And to give a better idea of that, we can look at how the system actually works when you're selecting the penetration locations and injection. So when you select this inject button, it brings up this window, which you can then start the injection procedure. And then using this crosshair, you can then move around to the different locations on these cells. So these are the cells. This round part here is the nucleus. And you can select all these different points. And then once you finish selecting the points, you hit enter. It goes through a path calculation routine. And then the traveling salesman algorithm calculates the shortest path between all these points to maximize the efficiency of the system. So eventually we would like to increase the speed of the injection once we have it working more robustly. And so the pipette travels to each cell, injects on an angle, and then the pump applies pressure to inject the cells. And so we've got the basic system now, so our next step is working towards incorporating our detection scheme. So with this penetration detection scheme, we wanted a good biological application, specifically when you're applying for grants and things to the NIH. You always want to team up with someone who has a good application for it. So we started this collaboration with Barry Cooperman and Ian Farrell in chemistry. So they generate fluorescent tRNA. So most people know that DNA encodes proteins, but the way that the DNA translates from a code on the DNA to a protein is using tRNA. So the mRNA from the nucleus goes out into the cytoplasm. It ends up on the ribosome, and the mRNA has the complementary code for the protein you're encoding. The tRNA is then has an anticodon that matches the codon on the mRNA, and brings peptides to the ribosome. So it basically translates the code here to an actual peptide, and then the peptide chain gets made on the ribosome. So that's how translation works. That's how cells produce proteins. And Barry Cooperman's group labels these tRNA with fluorescent labels. And he's done a lot of very cool studies, for example, looking at two complementary labeled tRNA on a ribosome. And you can actually get a fret signal due to the interaction between the fluorophores that lets you look at protein translation in real time. So why would we want to microinject fluorescent tRNA? So currently, they use lipofection. They collaborate with a lot of groups that use lipofection, which introduces this tRNA into all cells. So that's nice, but at the same time, it also takes several weeks to months to optimize the procedure to introduce tRNA into these cells. So with microinjection, there's no need for parameter optimization. We also preserve the composition of fluid we're introducing. So if we wanted to have tRNA with some other things, we can very easily change that. We can perform dose response studies. Like I mentioned, the injection fluid, the injection volume is linear with both pressure and the injection time. So you can easily inject, say, a group of cells at one rate, a group of cells at another rate, and then look at what happens at different doses. It has single cell resolution, and then we can inject into either the cytoplasm or nucleus, which is very nice because a lot of people are interested in the trafficking of tRNA across the nuclear membrane. So everyone used to think that tRNA was made in the nucleus and then leaves into the cytoplasm and doesn't go back. It turns out that the transport is bidirectional, so it goes both out of the nucleus and into the nucleus, and there's various modifications that occur with these different steps. So it's a very active research topic, it's a very hot topic, and no one else has these capabilities. It also gives us very nice time resolution for these studies. So if you transfect cells with other methods, a lot of times you have to let them incubate for a few hours. Here we can introduce fluid and then have a very distinct time equals 0 point. So we get very good time studies with this technique. So with that, we can look at tRNA trafficking dynamics. So these are just HeLa cells that we introduced some Cy5 tRNA into. And initially, you can see that there's almost no tRNA in the nucleus. In fact, some of this residual fluorescence could actually be just the envelope around the nucleus fluorescing. And then over a time of about 30 minutes, we actually see the tRNA migrates into the nucleus over time. And so with these videos, very simple fluorescence technique, we can then look at the trafficking data over time. And you can look at things like the tRNA fraction, so of the total fluorescence, what fraction is in the cytoplasm and what fraction is in the nucleus. And here we can see it almost doubles over time, so here's about 10% to about 20%. We could also look at things like the ratio between these the mean intensity. And from this data, we can then fit time constants to this data. So for example, with these cells, it was on the order of about a half-hour time constant for equilibrating the concentrations in the cytoplasm versus nucleus. So it's a very powerful technique for studying tRNA trafficking, and then we can then look at various factors that might affect the time constant of this trafficking. It also leads to some interesting observations. For example, here we have a cell that we injected, and a lot of times injection is kind of a rough technique. You poke a cell if there's vibration in the pipette, or if you overinject, the cell starts to die. So you get cell death due to either necrosis or apoptosis. And what we found was that during cell death, you actually get a rapid accumulation of tRNA in the nucleus of the cell. And maybe I can play that video again. So it starts out fairly evenly distributed throughout the cytoplasm, and then over time you'll see a very sharp increase in the fluorescence in the nucleus of the cell. Right around there. So there's a very clear accumulation, and we can see all the various morphology changes associated with cell death, such as membrane blebbing and nucleus kind of contracting, which indicates that the cell is in fact dying. And that becomes very interesting because tRNA has a known interaction with cytochrome C, which is involved in both cell respiration, producing energy aerobically, and also with cell death. So it's normally in the mitochondria, and then it is part of a cycle to produce energy for the cell. But when cell death is induced, it gets released from the mitochondria and triggers a chain that causes cell death. And tRNA is known to bind to cytochrome C, and if you introduce tRNA into cells, there's a statistically significant decrease in apoptosis. And so perhaps this movement of tRNA into the nucleus is some mechanism to prevent this interaction during cell death. It's also very interesting that there is this interaction because of the Warburg effect, which is where cancer cells predominantly produce energy not through aerobic respiration, but by glycolysis, which is an anaerobic method of producing energy. And why that's interesting is because in cancer and tumor cells, there can be up to a 3 to a 10-fold increase in the amount of tRNA in these cells. And so if you have this drastic increase in tRNA due to this increase in protein synthesis in these cancer cells, it could potentially be related to this tRNA basically impeding the ability of cytochrome C to participate in aerobic cell respiration. So this could potentially be one explanation for the Warburg effect in cancer cells. So with that, we kind of had this very interesting biological question that we're kind of started looking into very intensely recently. But another application that we have for CNPs is in microelectrode techniques. So at this point, I'd like to kind of shift gears a bit and discuss microelectrode techniques with CNPs. So microelectrodes are very nice because you get a less significant IR potential drop in the solution, which is due to the solution resistance. We get a faster time response due to the much smaller size that has a smaller capacitance. There's a smaller system perturbation, so you get you don't deplete concentrations in the solution as drastically. We get improved signal to noise and a much more localized measurement. So these are all the reasons why people kind of move towards microelectrodes. The benefits of CNPs as microelectrodes, as I briefly discussed earlier, is that we have very tunable geometry. So the tip size, taper, and amount of exposed carbon are tunable. And we can range from tip sizes in the tens of nanometer range all the way up to exposed lengths on the order of hundreds of microns. So we have a very wide range. We have batch fabrication. So instead of producing these one by hand, we can fabricate hundreds concurrently. And it's a very scalable process. So this could be scaled up quite easily. And then there's also simple electrical contact. So you can basically stick a wire into the back of the CNP. It contacts the carbon lining. So that's quite easy at the macro scale. There's no clean room required for fabrication, and it's compatible with all sorts of micropipette fittings and manipulators. So one of the first things we wanted to do is see that if we could detect cell penetration using CNPs as nanoelectrodes. So in fact, we can. So using the same technique, just with a CNP that's pressurized to the point where there's no fluid in it, we can monitor the capacitance signal as it penetrates the cell. And if you notice, the magnitude of the signal change using CNPs as these nanoelectrodes is much smaller. So before we were looking at a few picofarads signal change. Here we're looking at tens of femtofarads. So it's about 3 orders of magnitude difference in signal change. But with the lock-in amplifier, we have excellent time resolution and excellent resolution of this technique. So we can actually detect these changes. And again, we get a significant difference between penetrating into the cytoplasm or the nucleus of these cells. So again, it's on the order of a 1.5 times greater signal change injecting or penetrating or probing the nucleus. And this is just a blow up of this trace showing that it's a very repeatable process. You may also wonder what this peak right here is. So if you fit a time constant to this transient right here, you get about 0.3 seconds, which perfectly matches the literature value for relaxation time of the cell membrane. So we believe it's both due to relaxation of the cell membrane. There could also be a Cottrell current effect due to a jump in membrane potential across the cell membrane. But we can detect cell penetration. We can look at both the capacitance and real part of the impedance. And then if you're looking to automate this process, we can then look at things like the time derivative of the data, where in the time derivative, you'll get these spikes corresponding to large changes. And so you could potentially set a threshold value that is related to the downward motion of the pipette to detect penetration. Also, at the nanoelectrode scale, the pipettes are much more sensitive, and so we can actually look at the dependence on penetration depth. So here we had a CNP that we programmed to traverse downward at a constant rate, and we measured these signals again, the real part of the impedance change and the capacitance change. And we can fit this with a circuit model, and it's basically a monotonic dependence. So we can correlate the signal change that we're getting with the depth that we're actually penetrating the cell at a submicron scale. We can also compare that to data from a filled pipette, such as the microinjection pipettes that we had earlier, and show that this is actually the signal multiplied by 0.1, so it's 1/10 the scale. And you can tell that the signal is much, much greater when you have fluid inside the pipette, basically confirming our previous analysis. So that's one microelectrode application of CNPs. We also wanted to look at basically the most commonly used method of characterizing electrodes, which is cyclic voltammetry. So CV is a form of linear sweep voltammetry where you apply a linear increasing voltage to a certain rate, and then you reverse the scan in the opposite direction. And basically what happens is you get, as you increase the potential, you hit a point where a redox reaction occurs, and you get an increase in Faraday's current due to the redox reaction. And then you eventually hit a point where it becomes diffusion limited, so the peak dies off even though you're increasing potential. And then as you scan back the other way, you get the cathodic reduction current due to the reduction of the Faradaic species. And based on the location and the magnitude of these peaks, you can tell various things about the kinetics and the nature of your system. So here's just a quick characterization we did looking at cyclic voltammetry with CNPs. These plots are the current versus potential at different scan rates, so going from 10 millivolts per second all the way up to 10 volts per second. And in the microelectrode regime, you typically, under most normal scan rates, get almost a steady state current response. So there's not a huge difference in any of these curves. And we do recover a classic sigmoidal shape, which is characteristic of microelectrode cyclic voltammetry. And we can also look at the maximum current versus concentration. And typically the current in CV is proportional to concentration. And one application of cyclic voltammetry with CNPs comes back to detection of neurotransmitters. So we found we were looking through a catalog and found that carbon electrodes are commercially available, so we got interested in what they're used for. And it turns out that a lot of people use them for electrochemically detecting neurotransmitters, such as dopamine, serotonin, epinephrine, norepinephrine, adenosine. I think octopamine is also redox active. And they use methods, specifically amperometry, where they hold the electrode at a fixed potential and measure peaks in the current, or spikes in the current due to the presence of these neurotransmitters. Or fast scan cyclic voltammetry, where they apply cyclic voltammetry at a very fast rate to monitor these effects in real time. So to further elaborate on fast scan cyclic voltammetry, so it's at a very high scan rate, so we're talking about 400 volts per second, and they apply these triangle waves at a rate of about 10 hertz and measure the current response. And then they do a background subtraction to get rid of the capacitive charging currents that happen at high scan rates. And what it gives you is a differential measurement that allows you to detect these concentrations of neurotransmitters at a very high temporal and concentration resolution. So this is a typical cyclic voltammogram, or a fast scan cyclic voltammogram, where the current is the pseudocolor. So red would be a positive current, blue would be a negative current. These streaks correspond to the oxidation and reduction of a neurotransmitter dopamine. The y-axis is the potential that we're scanning. So it actually is a triangle wave, so it goes from negative 400 millivolts to 1 volt and then back down. So we're essentially taking the potential, plotting it on the vertical axis, the time on the horizontal axis, and pseudo-coloring the current. So these streaks correspond to dopamine. And the location of these peaks is how you can indicate that it's dopamine. Typically the peak location is dependent on the kinetics and the nature of the reaction. And you can look at the current at the peak. So you can take basically a line across this cross-section, look at the current over time, and you can get basically a map of the concentration over time. So this was with the CNP in a flow cell, where we, at about 10 seconds, turned on the dopamine flow. And then it ramps up and then hits some steady state concentration. And then at around 25 seconds, we turned it off, and it starts to drop back down. So you can do real-time measurement of neurotransmitter concentrations for cellular studies from a very localized region of cells. And with this, we decided to start up a collaboration. So we were not experts in fast scan cyclic voltammetry. So we got in contact with the Venton lab at UVA. So Jill Venton, if you're familiar with fast scan at all, is a progeny of Mark Whiteman, who was basically the founder of this technique and one of the biggest names in the field. And she's at UVA, and she's very interested in using electrodes to develop Drosophila melanogaster as a model organism for neurotransmitter research. So typically when people are studying neurotransmitters, they use mice as a model organism. So in order to do these studies, you have to kill the mouse, dissect its brain, take slices of its brain, and then use these brain slices in a very inconvenient and not pleasant way to study neurotransmitter release. It's also very difficult to upkeep or not difficult, but it's more expensive to upkeep mice, and they're very difficult to genetically modify if you wanted to look at introducing various characteristics of the mice genetically. Whereas Drosophila, they have a very short life cycle, a very cheap upkeep, and there's a lot of very easy ways to genetically modify them. So there is a need, in this case, for smaller, sharper electrodes to penetrate the fly's glial sheath. So they have this sheath around their brain that's kind of tough to penetrate. And with these carbon fiber microelectrodes, they're on the order of a few microns, so typically 5 to 10 microns. The fly brain region of interest is on the order of about 35 microns. So you're looking at a very large, a relatively large electrode trying to take these measurements, which they have done. But it would be nice if they had smaller, sharper electrodes that can penetrate the glial sheath of these flies and take these measurements, which got us very excited for CNPs. So we started this collaboration, sent them some electrodes. They started characterizing them. So there's a quick comparison between the size scale of carbon fiber versus a carbon nanopipette. This is what the background current looks like when you initially take the measurement. So you have a very large capacitative charging current. But after you do the background subtraction, you get very clear redox peaks corresponding to the oxidation and reduction of the dopamine. And you can look at the current signal over time corresponding to this characterization. We can also look at the dependence of the peak current on scan rate. So linear dependence is typically indicative of adsorption-controlled behavior, which we find with dopamine. So that's well classified with carbon fiber. And the peak current versus concentration, which we'd expect to be linear. So that's important for converting between peak current and concentration if you're trying to measure concentrations. They did some waveform optimization, basically just looking at two different ranges of potential scanning, so going up to 1 volt or up to 1.3 volts. They showed that 1.3 is both more stable over time and has a time signature that's very similar to carbon fiber microelectrodes. So this is just comparing the black is carbon fiber, the red is the CNP. We also noticed that with the CNP you get faster kinetics, which is shown by the peaks being closer together. And so with that, they moved to actual studies in flies. So here they use exogenously applied dopamine. So they actually stick a carbon nanopipette into a larval ventral nerve cord. And then they, using a separate pipette, apply dopamine. And why this is interesting is because you can actually measure how fast the dopamine gets uptaken by the flies. So typically when neurotransmitters are released from neurons, the dopamine that is now in the extracellular solution gets reuptaken by these cells. And so you can look at the uptake dynamics of the dopamine. If you didn't have the cells there to uptake the dopamine, this fast scan cyclic voltammogram would have a continuous streak across this way. So we can look at those kind of dynamics. And we can also look at the ex vivo detection of dopamine in optogenetically modified Drosophila. So in this case, again, Drosophila are nice because you can genetically modify them. So in this case, they genetically modified these flies to respond to a red laser or red light. So whenever you shine a red light on them, they release dopamine. So it's a relatively new technique, and they have these larvae that have these ion channels that are that respond to this red light. And so they actually stuck a CNP in these nerve cords of these larval flies, shined a red light, and you're able to measure dopamine concentrations in real time. So basically we basically characterized and demonstrated direct application of the CNPs for measuring these neurotransmitters in flies. And why we might be interested in the neurotransmitters are to study various diseases or various drugs that are associated with deficiencies or excess of neurotransmitters. So it's very promising work and a very nice collaboration with their lab. So in conclusion, we successfully developed a robust AC technique for detecting cellular penetration both during microinjection and cellular probing, even when the CNP is at the nanoscale. We've begun developing an automated system using standard software and microinjection equipment. And we developed and characterized CNPEs, or CNP electrodes, for fast scan cyclic voltammetry of neurotransmitters, specifically in Drosophila melanogaster. We demonstrated metallization and basic electrochemical measurements showing kind of the versatility of the carbon electrodes and how we can functionalize them for different purposes. And we used our microinjection expertise to develop new techniques to study tRNA trafficking across the cell membrane, or across the nuclear membrane. So the future work that we're looking to do, we're moving forward with the tRNA studies. So we would very much like to look at tRNA trafficking in response to various factors, such as amino acid deprivation, translation inhibitors, which prevent translation in the ribosome, cell death, which is something that we've discovered affects the tRNA distribution in cells, and heat shock. And we'd also like to look more into the interaction between cytochrome C and tRNA using our microinjection techniques. With the injection system, we're looking to incorporate the AC detection scheme into the Matlab GUI to make it a more robust system. And then for the fast scan, we're moving towards smaller CNP electrodes. So the larger electrodes have a larger current magnitude, but it's also a less localized measurement. So we're working towards smaller sizes, and we're also looking at attempting measurements in adult flies. So right now, those measurements are all in larval flies. But with adult flies, you can actually start looking at studying behavior of the flies in response to various factors. So with that, I would like to acknowledge everyone who's helped me along the way. It's been quite a lot of them. So of course, my lab mates in the Bau and Hu lab, the MEAM department staff, our collaboration with the Cooperman lab in chemistry, the Venton lab at UVA. We have a bunch of collaborators at Drexel who are also working with carbon nanopipettes, specifically Riju Singhal, Yuri Gagotsi, Gary Friedman. Michael Charlou actually was at Drexel briefly. He's now a professor at RIT, and he did his PhD work here for studying CNPs. We had some collaborators at Temple, Department of Pharmacology, and then also collaborators at Penn who have helped out with cell culture and equipment, specifically the Baumgart lab, the Mokowski lab, and the Meany lab. So with that, thank you very much. Any questions?